Hello Ji, Namaskar. I'm Dr. Nutan Prakash Vishwakarma, and today I am going to discuss a very important concepts of phylogenetic analysis, which is very important in bioinformatics. So, as you know, that since the starting of the civilization of the human being, the human was very curious, and that our curiosity and our intellectuality makes us different from other organism. All right. So, human always want to know what happened actually in the past, how the formation of humans takes place, right? How is the origin of life takes place, like that. So phylogenetic analysis is nothing, just it is the study of evolutionary history of living organism. So the scientists who are indulge in studying the evolutionary history of organisms, they have to go for the phylogenetic analysis. And the result they can represent in the tree-like diagram, and in that particular diagram you can the diagram represents the pedigree of those organisms. So the tree branching pattern representing the evolutionary divergences are simply referred as phylogeny. So what are the different methods available for the phylogenetic analysis? Although a lot of methods, the previous attempts by the scientists, many attempts were there, but here I am telling you the very simple one, which the first one, and people are using frequently right now. I'm not including all those methods because now we don't use them. So the first method is simply known as fossil. Fossil, you know that the fossils may be the fossil for the plant or fossil for the animals. So it is often studying using the phylogenetic analysis or phylogenetic relationship or evolutionary relationship can be studied through the fossil because fossil contain morphological information about ancestor of current species and the timeline of divergences. So as by the geological survey, they dig the mountains, the rocks, the somewhere, and they try to find out some fossils. If they try to analyze the anatomy or morphology of the fossils, scientists can correlate the two were the ancestor sequence, ancestor organism who were the descendant organism. So things are simple, but do you think so? It is possible for all the organisms? No, not at all. The fossil for specifically the fossil for microorganisms like bacteria, like fungus, like viruses, that is not available. So if any scientist want to study about the evolutionary relationship among the organism, so definitely there will be gap. The fossils for all the organism may or may not be available. So things is going to be incomplete. So this is not a very good idea to study the evolutionary relationship, although we can take the support of it, but to be dependent over it for fossil on the fossil for the phylogenetic analysis is not a good idea at all. So to study the evolutionary relationship by help of the fossils makes it impossible to study phylogeny with this approach. So now the most advanced method which we are using currently, that is known as molecular data, the molecular fossil. Why we call it as the molecular fossil? Because since the starting of the uh, existence of the cell, there was the genome, right? Maybe it is previously it was RNA and then it is uh, for the DNA because RNA was not stable. So 1 lakhs of years before, the cells had the DNA and still it had the DNA. Alright. And due to time, there is the during the time as time passes, there is the mutation. There is a lot of mutations accumulated in the gene sequence. So whatever sequence we have right now, that is much more changed as since the starting sequence. So what you can say, this is actually since our gene or DNA have accumulated the mutations, so we can call it as a molecular fossils. So like DNA and our protein sequences can also provide very useful evolutionary perspective of existing organism because the genetic materials accumulate mutations over time causing phenotypic changes. Alright, as mutation accumulated, it may change the phenotype as well. So genes are the medium for recording the accumulated mutation and they can serve as molecular fossils. So as a molecular fossil, we can use the DNA, we can use the protein, both can be used. So through comparative analysis of the molecular fossils like DNA, RNA, or protein from a number of related organisms allow us to follow the evolutionary history of the genes and even as a whole organism. I'll let you know. So the advantage of using molecular data or molecular fossils is number one, molecular data are more numerous and easier to obtain because you know that in these days now sequencing is very common. So molecular data or sequencing data you can easily now a lot of databases are there. We have discussed in detail in the theory session of bioinformatics. A lot of biological databases are available. So all the data is available on your single click. You can download the data. And even if for any particular organism you have to go for the sequencing, that is not a very difficult task. Simply you can go for the sequencing. So it is easier to obtain the molecular fossils or molecular data. Maybe it is DNA, RNA, protein. Mostly we use the DNA and protein. And there is no sampling bias involved, which helps to patch the gap in real fossil record. So as I discussed about the fossil, in fossil I told you that a few of the fossils for few of the organism only available. But once you are focusing yourself for phylogenetic analysis for the molecular data, simply you can extract the DNA of any particular organism and you can go for the sequencing. So there is no kind of the sampling bias. Point number three, more clear-cut and robust phylogenetic tree can be constructed with the molecular data. So therefore, they become favorite and sometimes the only information available to reconstruct the evolutionary history. So whatever we are doing phylogenetic analysis currently, that is totally dependent over the molecular data. The advent of the genomic era with tremendous amount of molecular sequence data has led to the rapid development of molecular phylogenetic analysis. As time will pass, sequencing will be cheaper. More data will come to uh, databases by scientific community, and we will be in better position for the evolutionary relations of study for all the organisms in coming days. So simply the phylogeny, which is dependent over the molecular data, that is simply known as molecular phylogenetics. So the study of evolutionary relationship of genes and other biological micromolecules by analyzing mutations at various positions in their sequences and developing hypotheses about the evolutionary relatedness of biomolecule based on the sequence similarity. So based on the sequences, we do study about their mutations and similarities, and based on that, we develop the hypothesis about the evolutionary relationship among the organism. So what are the various assumptions while we are dealing with the reconstructing the evolutionary history by using molecular data? So there is number of reasonable assumptions. Number one, few assumptions we have to make. Number one, the molecular sequences used in phylogenetic constructions are homologous. Means whatever sequences we are using, they are homologous. Homologous means very simple. Like they show their ancestors were same. They show the very strong level of similarity. Meaning that they share a common origin and subsequently diverged through time. And during the course of time, they were diverged. Previously, they were from the same ancestor, and as time passes, sequences become diversified. Point number two, the phylogenetic divergence is assumed to be bifurcating, means from a single ancestor there were two descendants only, meaning that a parent branch is split into two dotted branches at any given point. Assumption number 3. The variability among sequences is sufficiently informative for constructing unambiguous phylogenetic tree. Means whatever variability is there among the sequences that give the information for constructing the phylogenetic tree. So phylogeny or species, you can say the phylogenetic tree, or you can say the species tree, or you can say the evolutionary tree, represent the evolutionary relationship among a set of organism or group of organism. And the group of organism whose ancestors are same or common, they're simply going to be called as a taxa. All right. We'll let you know while we will dealing with the terminologies of the uh, phylogenetic tree. Then you will better understand the taxa. So what phylogenetic tree is all about? So phylogenetic tree is an illustration of the evolutionary relationship among a group of organisms. Very simple. There is two term. The one is known as gene tree and species tree. So what gene tree is all about? Gene tree represent evolutionary relationship of a particular biological molecule, like gene or protein product among species. Say, suppose you have one gene. The name of the gene is known as TP53, and you want to study about the evolutionary relationship bit among the TP53 of different organisms. So what you will do? You will just sequence the TP53 genes of all the organisms which you are involving for your phylogenetic analysis, and at the end, whatever the phylogenetic tree will get, that is going to be called as a gene tree. So that tree will represent the evolutionary relationship in the TP53 genes of different organism, right? So that is the evolutionary history of a particular gene, not for an organism. Say for example, suppose in the phylogenetic analysis, you find that TP53 gene of the human and mouse is their ancestor is common. It doesn't mean that as an organism, human have the close relationship with the mouse or something else. So gene tree is different and species tree is different. Like hemoglobin tree, kinase tree. So if you make the hemoglobin gene tree for the hemoglobin, that will represent the evolutionary history of hemoglobin only. Kinase tree, a protein. So that will represent the evolutionary relationship among the kinase protein of different organisms. You can't say that these two organisms are closer. Few of the students are always get the confusion about the guide tree. Once you do the multiple sequence alignment by the cluster W or cluster omega or any family of the cluster, you will find the tree like structure, and that is people understand that it is phylogenetic tree. No, it's not like that. That is simply known as guide tree. We'll discuss about the guide tree while I will make the video on the multiple sequence alignment. So guide tree produced during multiple sequence alignment have no phylogenetic meaning. So the dendrogram are based on distance derived from pairwise alignments. They are used to determine in what order sequence are aligned during the construction of the MSA. So as of now, I'm not discussing much more about the guide tree. Just simply notice that guide tree is related with the multiple sequence alignment. Alright, that don't have any relationship at all with the phylogenetic tree. Although it looks like the phylogenetic tree, but that is actually the guide tree in case of multiple sequence alignment. Here you see the gene tree and species tree. I give the example. Suppose you have the gene A, B, C, D, E something, uh, and here you have made the phylogenetic tree. So here the tree represents the evolutionary relationship of the tree irrespective to the organism. The two event mutation because different genes formed due to the mutation, and species formed due to the speciation. So here you see the species A, B, C, D, E. So speciation events happen, then different species form. So gene tree is different and species tree is different. Whatever results you are getting in the gene that you can't impose for an organism. All right. So the two event mutation and speciation are not expected to occur at the same time. So gene tree cannot represent a species tree. So who use phylogenetic analysis? What are the benefit? What are the applications of phylogenetics? in different sectors. Number one, evolutionary biologist, the scientist who involve in the study of the evolution relationship, means they are trying to make the reconstructing the tree of life. They use the phylogenetic analysis. The scientists who involve in the systematic, means classification of organisms, they also use the phylogenetics. Anthropologist means origin of human populations, and that there is also applications of phylogenetic. Forensic science, forensic science scientists also can use the phylogenetic analysis. They can track the transmission of HIV virus to a rape victim, and that can be used as a proof also in the court that if someone have raped any. So maybe if the rapist have the HIV, that HIV strain reach in the victim, so that can be proved in court. Parasitologist, like phylogeny of parasites and coevolution. Epidemiologist, like a reconstruction of disease transmission. Say, for example, any pandemic that takes place and that is due to virus. So from which country actually that virus is spreaded, that can be tracked by the epidemiologist by using the phylogeny. Genomics and proteomics means whenever you have to compare the homology of new proteins or new genes, you can use the genomics. The person involved in the study of genomics and proteomics, they also use the phylogeny. So these were the very basic detail of the phylogeny. And what are the applications of phylogeny? We'll see you in next video, and then we'll discuss bit detail about the phylogenetic analysis. Till then, take care. See you. Jai Hind.